Thank you very much, Dr. Shao, and I'd like to thank SINOVAC and Co-Benefit for the invitation to present our work. So I will share my screen now. Is it good? Can you see my slide?
Yes.
Great. Thank you so much. Good. So I'd like to just remind how pandemics have been affecting humans for many centuries, and the work that science does in a collaborative manner is important to develop vaccines and other approaches that could help to control the pandemic or the spread of infections. So that was the case for smallpox, a virus that was eradicated in the late 70s thanks to the work of science and public health worldwide. And that's been also the case for other diseases that are due to viral infections such as polio, measles, rubella, and mumps. So every time a vaccine is introduced in the population, there is a clear, when the vaccine is safe and effective, then you see this reduction in the numbers of cases in the population. So in the overall, vaccines are successful tools. They can lead to a decrease in the number of cases for several different types of diseases. And as long as the coverage of the vaccine goes up, the protection in the general population increases. So one of the things that we need to confront, particularly from Chile and other countries, is the gap between what we call industrialized countries and developing countries, where the number of vaccines and the access to vaccines and their specificity of vaccines might be different. So it's important that we try to fill this gap. And we published a paper in 2018 showing that those countries that produce vaccines have usually more successful vaccination campaigns or vaccination. The impact of vaccines is better in those countries that have their own manufacturing capacity. So in our region, places like Brazil, Argentina, Mexico, and Cuba are the only ones able of making vaccines. And Latin America is a huge part of the world in terms of the size of the region and the people. So in collaboration with SINOVAC, we from the Católica University, we are trying to reestablish a capacity that Chile had in the late 90s, and that's saying to produce vaccines here. And these projects, we've been able to collaborate with SINOVAC in terms of just promoting, facilitating the proper landing of SINOVAC in Chile. And so far it's actually moving very well. Now, when confronting the pandemic with the need of a vaccine, we realized early after the pandemic was initiated that research and collaborations leading to vaccines for the SARS-CoV-2 are very important. And we initiated a study that published in Frontiers in Immunology in May 2020, showing that our design of a vaccine based on BCG might have a chance, because countries that use BCG as a vaccine for tuberculosis, they have a reduced rates of outbreaks as compared to those countries that do not have a BCG program. So we are currently still working on this vaccine. We think that the quality of BCG, of inducing and trained immunity response in humans, might be beneficial to prevent the inflammation that is is one of the main causes of the severe cases of SARS-CoV-2 infection. So we think that our approach has some potential advantages that we are actually currently studying. So we been working all through the pandemic since January 2020 on trying moving forward with a prototype that was designed by us at the Catholic University in Chile. These are the students and postdocs working on that. But at the same time, I mean, this work that we've done, which is just scientific work based in Chile, had led us to was highlighted in important places like Nature. In this article, which I find very nice, Latin American scientists joined the coronavirus vaccine race because no one's coming to rescue us. And also in New York Times, this is a coronavirus vaccine tracker where our work's been mentioned. And of course, that led to, before the pandemic, we've been working on vaccines for a couple of decades up to now. And we initiated before the pandemic an interaction with scientists and collaborators in China in order to work on vaccines. And one of the main events was actually in, I think it was September, October 2019, as part of the International Congress of Immunology that took place in Beijing, where we had the chance to present our work and also learn of the work that SINOVAC was doing on vaccines. So we were planning to collaborate, and then the pandemic came, and SINOVAC developed advanced development of this inactivated virus vaccine that was published in Science. It's an approach that we appreciated from Chile because first, it's safe, it's known to be safe. Second, it could be used in all ages. Third, it has all the antigens from the virus. And finally, for a country like Chile, a vaccine that requires 2 to 8 degrees centigrade is easy to handle, and the country has experience dealing with such vaccines. So then we were, of course, in touch with very high frequency, and we knew about the study that was published in Science, and of course we knew about the clinical trials that were in progress, both in adults and elderly, that led to their publications in Lancet Infectious Diseases. And knowing this data in advance, of course, encouraged us to initiate a collaboration specifically on COVID-19, on top of what we were originally planning to do. And so we created a MOU and then several agreements that were supported also by the governments. And from our side, from the side of the university, together with SINOVAC, of course, we work on a clinical study protocol that was submitted to the ethical committees and the Chilean regulatory agency that is called ISP. That was approved in September 2020 and led to the registration of the trial in clinicaltrials.gov. This trial involved 8 different centers, different parts of the country, and has a central scientific lab that was doing all the immunogenicity analysis. Different places of Chile were participating in this trial. And in terms of safety, data were quite encouraging. So the vaccine leads to just to minor side effects. Most of them are pain at the site of injection, some headaches, but not really major side effects, such as we have non-anaphylactic responses, for example, as part of the trial. And we focused on a very well analyzed immune response, showing that the antibody titers become significant 2 weeks after the second dose, and that the second dose was really needed. And as we will see now, we're talking about a third dose or a booster dose. And equivalent results we obtained from the total people enrolled in study, as well as elderly. And this is just the early data that was we submitted very quickly to meta-archives, but it was accepted in Clinical Infectious Disease, a high impact journal. We did several tests to evaluate neutralization capacity of the antibodies. And again, we found that 2 weeks after second dose, a significant neutralizing capacity is observed in the subjects that were enrolled in the study. And this was, we were able to look and differentiate the immune response, of course, between adults and elderly. And this was very important data that were analyzed not only by the Chilean MOA, Ministry of Health, but also by other countries in the region. And we found that there was a significant increase in the neutralizing capacity of antibodies over time, both in adults and elderly. So the and this is just more data showing the same concept, different tests for we did direct neutralizing of the actual infectious virus, but also we used surrogate kits. We use a pseudovirus, and as I said, we had 4 different strategies for looking at neutralization capacity of antibodies induced by CoronaVac. But we also decided that it was very important to look at the immune response, at the cellular immune response triggered by the vaccine. So in collaboration with colleagues from La Jolla in the US, we got some peptides that specifically prime CD4 T cells or CD8 T cells from people vaccinated. And these are megapool peptides that can bind to MIC class 1 or MIC class 2. So we can differentially evaluate by ELISpot the presence of cells that recognize these peptides from the different antigens from coronavirus. And we could see that after the second dose of CoronaVac, there are significant T cells that could be detected in the circulation of the subjects enrolled in the study. Also, we had a paper that was just published showing that we have a few cases of COVID-19 as part of the study. We got a rate of about 2.5% of cases. So we analyzed very carefully the immune response of the people that are these breakthrough cases as part of the study, and found that the immune response after exposure to coronavirus led to a very strong activation of both antibody and T cell mediated immunity specific for the coronavirus, suggesting that the memory, the immune memory was there, and it was working in people that got infected with the virus. And this is the data, and we were able to compare between 2 different immunization schedules in terms of the response that subjects that were exposed and infected, they were just infected from the coronavirus circulating in the community, were actually responding properly in terms of this memory recall response after exposure to the virus. And we did the same with T cell responses, and now we are correlating the clinical outcome of these patients with their antibody and T cell levels against coronavirus. And we, as I said, we just published this paper a couple of months ago. So as part of this very virtuous collaboration, we have published now, I think so far like 4 papers, and there are 2 of them under review. We have as preliminary conclusions that the vaccine has been well tolerated, and especially in the elderly. We have not severe adverse effects or withdrawals, no anaphylaxis from the study, and a very well characterized immune response after immunization. And we know that the antibodies are produced and can be detectable at high levels 2 weeks after second immunization, second dose, and also we are able to see and detect very clearly T lymphocytes that produce gamma interferon. Now, this has enhanced our capacities in Chile, so I think we're very we're confident that the capacities that we have built in terms of teamwork, protocols, lab protocols, equipment has actually improved our capacity to do research on vaccines. And of course, we've been working all along through SINOVAC, just connected to what was taking place in Brazil, Turkey, and Indonesia. Now, most importantly, as part of this collaboration, besides of the science and the clinical studies, it was the connection between China and Chile. We got very strong support from the government and a foundation in Chile that led to this clinical and scientific trial that allowed the country to access to doses required for conducting a mass vaccination program. So based on the data from all the trials that were going on CoronaVac, plus our data in the trial in Chile, the Chilean regulatory agency and the Ministry of Health approved the vaccine in January 2021, leading to the first shipments of doses for the use of the vaccine in Chile. And this is very important because this is probably the first time that we have in the history ongoing scientific research, clinical trials, and mass vaccination at the same time in a country. So the connection with between SINOVAC and the Chilean government led to this access to the doses required for mass vaccination, and we start seeing some of the benefits of that process during March and April. And it was very impressive that the way the Chilean government, and just not only the government, but the local government, and also the small healthcare systems that is really well prepared in Chile for vaccination, led to a very efficient process of vaccination. And as you see here, most of the vaccines that have been used are from SINOVAC, and it's pretty amazing, I think, the way the vaccination campaign was able to control this major second wave that was taking place in the winter in Chile. Remember that we had winter during March and September. I mean, cold temperatures. The winter starts in July, but the low temperatures begin between March and April. And as you can see, the vaccination campaign was very contributed significantly to keeping down and controlling, reducing the number of cases of COVID-19 in the country. And this is thanks to the collaboration that we did with SINOVAC and the agreements that were signed between the university and SINOVAC and the government and SINOVAC. Now, the Chilean Ministry of Health did a effectiveness study that showed pretty clearly how effective the vaccine was after a couple of months of the vaccination campaign. So the lowest number is 67%. That's to prevent symptomatic COVID. But as you can see here, this effectiveness is a very encouraging effectiveness much higher, for example, than what you get with influenza vaccine, which is about 40%. Now, in terms of numbers of side effects out of the like population studies, in July this year, there have been several reports, but this is one of them showing that CoronaVac is one of the vaccines that has the lower number of side effects, and this is and it's been, you know, if you look at this number, it's 5,000, but it's because most people got SINOVAC here as compared to other vaccines, and this is the number that really matters. It's about 36, about 100,000 doses that were administered. So this is pretty low as compared to the other vaccines that were used here in Chile. So the main conclusions, and this is what we really are proud of, is that first, we helped to contribute data about safety and immunogenicity of SINOVAC vaccine, and we were able to characterize the immune response and provide data for the proper use of the vaccine in the population. But the fact that safety, immunogenicity, and efficacy data from the controlled trials are predicting what's going on the population, as a scientist, to us, a scientist, for us, this is very motivating, very exciting. Now, some questions that we have already answered and the papers have been submitted is how long the immunity lasts, whether additional boosts are needed. We have already answered those questions. Capacity to neutralize variants. We also have that paper, I think was accepted. And also whether, and this is very important, the vaccine is safe, immunogenic, and efficacious in pediatric population. I just show you just a few data, and with this few slides are going to be ending. If we just look at the 014 or 028 schedule of vaccination, we were able to provide to the health authorities data showing that by 6 months after the first dose, there is a clear diminishment of antibody and T cells. That was sort of a sign of for recommending that an additional booster dose might be needed. We were able to show that the antibodies induced by CoronaVac immunization lead to cross-reaction with most of the variants of concerns. This is very important because we have no longer in Chile the original strain circulating. We have mostly Gamma and Delta. So having data supporting that the immunity induced by CoronaVac can neutralize those variants is important. And most importantly, we recently, and this is actually out already as a preprint published in meta-archives. I was actually posted yesterday. We were able to show a very significant increase of the antibody and T cell levels after a third dose of CoronaVac in people from the study, suggesting that in addition to the safety, the booster dose is very efficacious at increasing the levels of neutralizing antibodies. This was 6 months after the first dose. As I said, this data are already posted as a preprint in meta-archives. So one thing that's very important, countries that have moved efficiently at vaccinating the population have confronted this problem, which is shown here in this slide. The virus is starts looking, adapting to infecting people that have no immunity, and the pediatric population is of a special concern. And these data are from Israel, where you see that the frequency of pediatric population infected and sick because of COVID-19 is increasing because of the adult and elderly population vaccinated. And that's why we decided to move forward with a clinical study for pediatric population. We initiated the study quite a few weeks before the MOH decided to initiate a massive vaccination of children 6 years and above with CoronaVac, and these are the numbers for their vaccination campaign, which has been very active, and these are a little bit outdated, but these numbers are reaching now about 60%, 70%. So we are right now in the midst of this phase 3 trial that is multi-center with other countries, and this trial is going well. We already have the first safety and immunogenicity data that we share with the Chilean regulatory agency. They are particularly interested in the data between 3 and 5, which is the age group that is supposed to be next in terms of population vaccination. So this work was done combining basic research, clinical research, support from the public sector and the private sector. Of course, our collaboration with SINOVAC, international collaboration that goes across borders. And, but most importantly, I think, is the trust that citizens have on vaccines. And we have always used this approach, which is, I'd like to highlight this editorial from Nature showing that vaccine confidence requires radical transparency from science, and that's the way we've been handling our work on vaccine, as we did before for other studies we did on, for example, respiratory syncytial virus. So this is the core team working on the adult study, and I'm very thankful to them. They've been really a great team and work very hard. And of course, now for the pediatric study, which is larger, there are more people involved, and we got a very strong support from the university authorities, and most importantly, many scientists, clinicians, nurses have been critical for this work to be done successfully. So thank you very much. That's what I wanted to share today in this presentation.